This video is going to describe the structure and function of the kidney. So here we have a kidney cut in half, and in red there we have the renal artery bringing blood into the kidney, and you've also got the renal vein in blue, which is taking blood away from the kidney. We've also got another tube in here leaving the kidney, which is called the ureter, which is taking the filtered waste products from the blood away from the kidney to the bladder. Now the outside part of the kidney is called the cortex, and the inner part is known as the medulla. Now the kidney is made up of millions of structures called nephrons. I'll try to draw where they are in here. Okay, so there's millions of structures like this, starting in the cortex, going down into the medulla, back into the cortex, and then back into the medulla again. And these structures are called nephrons. So here I've drawn a diagram of a kidney nephron. One end of it, we've got what's called the Bowman's capsule. And then this first part here is called the proximal convoluted tubule. It then extends down into the loop of Henle. It will then go back up the ascending limb of the loop of Henle to the distal convoluted tubule, and here we have the collecting duct. Now blood will flow through what's called the afferent arteriole into a dense network of capillaries, kind of inside the Bowman's capsule, which is called the glomerulus. And then the blood vessels then leave the glomerulus via the efferent arteriole. Now, in the glomerulus, the blood is under very high pressure. The diameter of the afferent arteriole, so the blood vessel going into the glomerulus, is larger than the diameter of the efferent arteriole. That means lots of blood is going in, little blood is going out, therefore we've got a high pressure inside the glomerulus. This means substances are filtered out of the glomerulus into the Bowman's capsule. The substances that are filtered through into the Bowman's capsule are smaller molecules like water, glucose, amino acids, ions, charged particles, smaller proteins, and urea, which is the toxic waste product of breaking down proteins. The only things that remain in the blood are larger proteins and cells. So, inside the filtrate, in here, in the Bowman's capsule, we've got a lot of smaller molecules. Now, we say they have undergone ultrafiltration. So, these substances move down the proximal convoluted tubule, and as they go down the proximal convoluted tubule, anything useful is selectively reabsorbed. So this requires energy, and useful molecules like glucose and amino acids are reabsorbed back into the blood. So if we continue the efferent arteriole, the blood vessels actually kind of circulate round the nephron, really close to it, so substances can be reabsorbed back into the blood. So the filtrate will continue down the descending limb of the loop of Henle, then up the ascending loop of Henle, through the distal convoluted tubule, and then into the collecting duct. The filtrate will then travel down the collecting duct, which then travels to the ureter, which all the many collecting ducts from the millions of nephrons all converge into the ureter, which takes all this waste filtrate product to the bladder. Now this describes one of the main functions of the kidney, that it filters the blood and removes any waste products from it, such as urea. However, it also plays another important role. It's involved in controlling the water content of the blood. In other words, it's involved in osmoregulation. Now the hormone involved in osmoregulation is called ADH, antidiuretic hormone. Now this dotted line is representing the normal blood water content level. If we get a decrease in blood water content, this is detected by the hypothalamus, which is just a structure towards the base of the brain. The hypothalamus sends impulses to the pituitary gland, and the pituitary gland then releases ADH directly into the blood. Now ADH is a hormone and it affects the kidney. It actually affects the collecting duct wall within the kidney. It makes the collecting duct wall more permeable. This means that water is able to be moved from the collecting duct back into the blood. This means that if there's water moving from the collecting duct back into the blood, you're actually going to produce a smaller volume of more concentrated urine. And as water's being reabsorbed back into the blood, it's going to raise the blood water content levels. Now, oppositely, if there is an increase in blood water content, the hypothalamus once again detects this. It sends impulses to the pituitary gland. Now, on this occasion, the pituitary gland releases less ADH. If there's less ADH in the blood, the permeability of the collecting duct wall decreases. This means there is less water being moved from the collecting duct back into the blood. This is eventually going to lower the blood water content, and you would end up with a larger volume of dilute urine being produced. Now, this process of osmoregulation is an example of negative feedback. Negative feedback is just where a change in a factor leads to the opposite change happening in order to keep things fairly constant. So, for example, if there is an increase in blood water content, the pituitary gland releases less ADH, which then results in the blood water content levels decreasing. And oppositely, if the blood water content levels decrease, the pituitary gland releases ADH, which then results in the blood water content increasing again. So hopefully you've now got an understanding of the structure of the kidney, in particular the structure of the kidney nephron, and what goes on in the kidney nephron. You need to be aware of ultrafiltration, selective reabsorption, and also the role of ADH in osmoregulation.